the screen? Can you see the screen?
Yeah, I can see the screen.
Okay. So那个
浩然。
甄浩然 okay, okay. And so you asked me the basic course structure, and so I, as a introductory course, I show you now. And this is I just prepare for this slide size. Yes. It's okay, I guess. It's okay from the beginning. Yes. Okay. And then this first, our structure is community analysis of microbiota. And this is a course that was that I thought nearly 3 years ago. Yes, it is exactly 3 years ago. Yes. And and our teaching system is indeed it is yes, it is 3 years ago, and also in this semester in the spring semester that I at that time I took a course that is yes, and then they set a final project, and then I submitted this as the final presentation called Community Analysis of
哦，你
Microbiota.
家长休息，
Hello?
有空的教授。
什么？
哦，主要这个
What? Oh, well, it doesn't matter. I mean, you can use your airport.
year, but I need to say to you, speak with you.
Okay, okay, okay. It doesn't matter. Yes.
Okay, I found a better place.
Okay, it's okay, okay, okay,
Yeah,
okay.
let's continue.
Okay, and then the first is that this course, and we have currently we have four teaching assistants, and I mean, but it seems that they haven't finished their final exam yet, and they are yes, first is this Zhao Ruohui, and then Yan Rong is this, and then Lei Teng is this. Yes, I came 这个齐汉生 is not here, and I came last month, and then I have a visit to Chantal, and so I have a dinner with them. So we have a group picture. Yes, and they are from another. We are currently ongoing data bioinformatics project called diabetic neuropathy. And they do some very good data analysis job in that. No, not not yet, and they're doing the data processing, and they do a pretty good job. So they the teaching assistant in this course, and they will help you if you have any questions in such as any question, any kinds of especially some details about how to installing like those, because our course is a very intensive course. You can see the first is the besides some basic introduction. This our knowledge basic introduction. Our main focus is you need to learn a lot of bioinformatics software and languages. And then, for example, like Linux, some basic use browse and Python and R and Java, and you use those languages, and then you need to have. I mean, you do not need to master every language, but you need to at least know how to install those software and how to run the related software that we required. This is, and then we down, and then yes, and so there might be you might encounter many difficulties doing this procedure. So this is why we need for teaching assistants. I mean, I'm not sure maybe whether four is enough. I hope it is enough. If not enough, I can find more. Yes, and because I expected that you will have what is your major? You are majoring in biotechnology, right?
Yeah,
Oh, okay.
um,
You are majoring in biotechnology. Yes. Okay. Yes. Yes. Do you have what kind of courses you have took? Yeah. Are you a sophomore? Sophomore.
what is sophomore?
Is called freshman. What is your year? Year is first year, second year, third year, or fourth
Uh,
year?
first year,
First
CSE
year.
student.
Oh, you are in
Yeah,
the first year. Okay. Yes. And so you haven't took introduction any computer courses, right?
yeah, yeah.
You also you haven't took any computer course, and you whether you have previously in your high school did any related courses about that, like learning these languages or self-learning,
Actually, I know something about Python.
or you know something about Python.
Yeah.
Or what kind of your level is running some others programs, or you can what kind of levels?
Um, I can say in the last year, a very genius student take take us to have a competition.
Or
Yeah.
you join a competition, it is national level or province level or school level?
It's let me think how to say.
Yeah, city. Well, no, well, but if you join a competition, that means what is your competitors? You know, from where? If it is from the same school, that means it is a school level. If from the same
No,
city,
no,
it
no.
is the city level.
It's a country level or it's a world level. But I think it's just country level. It's
Or the country,
uh
or it is so it is a very big
yeah, yeah,
competition.
very big competition.
Or
But we
do you want any award in that competition?
um he because we have two teams and that guy the team uh he got he 我会用英说
You can say that. Try. Or you I mean you say he, but what is your? I mean I care about your.
me
Uh,
I
we.
actually
You say we. Oh,
we
okay.
okay we
Yes.
but however we have two teams and one team get a good grade and he in the next go to the next let me find the word final competition
But
and we just ended before it
okay.
and we lost
So you
the competition.
enter the final because in over competition there are many different levels. You will pass the first screen and then enter the final level, final one-to-one competition. Is my understand correct?
Uh, not not yet.
Not yet.
I
Okay. I understand. I mean, basically, so you have some. So I expected that you will at least you know many basic. You already learn many basic programming. Some basic such as how to write a very simple Python program. You know how to do that, right? Yes, and then you also know something basic about data structure or something like that, right? I expected that, yes, and that would be very good. I mean, if you can do very well in this course, and then you can help me do something, and then we have some other courses, the other projects that you can help us with, right? Yes,
okay
yes. You if yes, you can see that, and so that our data structure is like that. For example, we this is a course description. You can read it by yourself. So yes, and so this is why I said that there is no very strict prerequisite because I mean, it depends on you. It depends on you. If in your example, I would assume that because you took a national level exam, I would assume that you are similar to this after you study introduction to computing with Python or and the introduction to computer science. I would assume basic level similar to that. But you haven't studied microbiology and advanced genetic technology, right? Yes, and so I think that we will also basically introduce something about
we
the
just learn a little in biology one because about the biology course we just have two courses. One is biology one and the other is biochemistry enzyme.
oh,
Just these
and
two
then
courses.
biology one is Kain taught.
Uh, CBCA and time
Uh,
half half.
Another teacher and Kain.
Yeah. Another teacher and
Yes,
time.
of course, and he is a very famous microbiologist. And then yes, and then we have something like that. I will also briefly introduce some basic backgrounds in microbiome that is required necessary for our understanding. Yes, and because if I know your background, and then I can adjust it. And then after that, and then we will first introducing, for example, like, because the key thing that we are introducing here is that we are introducing a NGS sequencing next generation sequencing technologies. That is, we can this method can exhaust it. I mean, all the possible, nearly all possible species in such an environment. Yes, and for example, if we select 16S, 16S is for bacteria. I will introduce it later, but generally the main point of our course is this is a bioinformatics course that we are mainly focusing on how to analyzing the data generated from NGS. Yes, and there are, for example, the NGS sequencing in microbiome, the microbiota in our case, of course, it might also contain some virus, but we don't consider it in this level. We have generate this is 16S, 18S, and ITS, and is bacteria and fungi. Yes.
So
Uh,
will we operate some machine to do the next generation sequencing in this course?
sorry, no. We basically downloaded online data. Yes, we would
So it's
download online.
just
Yes.
data, it's information.
Yes. We
Mm.
download online data mainly from here. You can see is NIH. We would download online data. Michael.
Ah,
Yes, we would download online data. Yes, we can. Yes, for example, it depends. It depends, and for example, you can see that there are many microbiome data. Yes, like this. This maybe we can. I just randomly read that. Yes, randomly read that. There are something that sometimes there are some because you need to very carefully, yes, very carefully NGS data, or if you go to the yes, go to some microbiome, because microbiome have many other databases. It's many, yes,
yeah.
microbiome databases. Yes. Have many different databases or human microbiome projects. There are many databases that we will basically download the data from the databases and then use as examples, yes, to study. And we want, I mean, if because it don't have the time. I mean, if you try to use you go somewhere to gather some sample and then do the sequencing.
Yeah, yeah.
I mean that of course after you learn this course you can do that. Yes, you can go to other professors' lab, and I mean, I think they will be very welcome to you to go there. Yes, and then you can do some do the sampling and then sequencing. Normally, it takes more than 1 month. Yes, and I mean, we it is very impossible for us. I mean, we need to finish this course in about 2 weeks. Yes.
Okay, okay.
Yes. We will download online data and then as an example, and then we will as an example, and then here you we as an example we will first introducing you some basic things about Linux, Python, R, and Java. And as if I know your prior background, it will be easier. But there are someone if they are not very familiar, and then especially you are familiar with Python, you can teach them, right? Yes, you can help them. Yes, and then downloading, and then we learn how to downloading this sequencing data from online. Indeed, this processing is not as simple as you think. I mean, even downloading this data, I mean, at least previously we spent some time to do that. Yes, it's not that easy. Yes, especially it's not. Yes, it depends. It depends. Like the Earth environmental project. Yes, it depends. It depends on where you download and how good your network is, and then you also yes which platform you download. Yes, and then we briefly introduce that, and then this is some basic things, and then hey wait. Oh, and then after we downloading the sequencing data, we can do some, and then we will use them as an example to do some data analysis. And then the first is doing the microbiome diversity analysis. It is this it depends. They have many different many different algorithms. Like the basic thing is alpha diversity and beta diversity. But there are also some other diversity computational methods. And if you're using the CHIME tool, and then it will automatically generate it when processing the sequencing data. So, but we haven't decided whether use CHIME tool or use CHIME. or maybe we can talk more. We can try both. Yes, and charm two is much more popular, of course. And then this is taxonomy and phylogeny. Phylogeny. Do you know phylogeny? It's some phylogenetic tree. Do you know that?
No,
You don't know. Yes, you can. Yes, it is something like that. Yes, I think you'll read this in
yeah, yeah,
biology. Yes.
biology one.
Yes, 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 yes. It is some phylogenetic tree. So in this microbiome, after we analyzing, for example, from one sample from the human gut microbiome, or another from the like the guy in Lam, he done something like use from extracting the microbiome from cow to study antibacterial, right? Yes, and then all like Igal Akumon, Professor Igal Akumon, he do something like in extracting the soil microbiome, and then in the microbiome there are many bacteria. So normally the first step is that is generating a phylogenetic tree, uh, really to sort it all of this really the relation of all this bacteria, all different kinds of species, and this they have a computational algorithm, and I will introduce it. So yeah, yes, phylogenetic similarity. Yes, yes, they have a basic computational algorithms to study how we. I mean, we would very detailed discuss that how we generated this phylogenetic tree. I mean, the basic algorithm how it works. But of course, that if you're using the charm tool, it will automatically generate a phylogenetic tree for you. And I will introduce why they do that, why they use that, yes, and basic principles, and uh, and then it's functional analysis. And after we do, I mean, normally the first step is doing some phylogenetic, and then studying the microbial diversity, and then we will study the functional analysis. It is functional is something like that. We study the metabolic pathways within the microbial community. And it's like that. Yeah, but because the data we downloaded, in fact, it is a 16S data, 16S data. But the 16S data do you know 16S data?
Mm,
Yes.
it's
It is a
maybe I don't know.
ribosome. It is a ribosomal RNA. Ribosomal RNA.
Oh,
Yes, yes. And so this ribosomal RNA, and then it requires. I mean, so it is just like that. They extracted all the RNA within a sample. For example, no matter it is a soil or a gut vessel or anything else, and then extracted all the RNA, and then they use it some centrifugal, yes, to select it a certain proportion of RNA that is 16S. Yes, do you understand that?
can you repeat again? Maybe I.
Yes, yes. I mean, they extracted all the RNA within a sample.
Oh,
Yes, and then they using some methods to extract it a certain proportion of RNA within this all total RNA. And then this RNA is 16S ribosomal RNA is very unique to bacteria. I mean, if you are, for example, like we are our human, we don't have for animal, we don't have this 16S ribosomal RNA. This is unique to bacteria.
it's unique to bacteria or it's unique to unique
Generally unique
bacteria.
to bacteria.
Okay,
Yes.
generally.
Yes, 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 yes. And so and so that and so this 16S ribosomal is very good for identifying the bacteria. I mean, and more important, because we need to use the ribosomal RNA because you need to understand that in biology one we say that the ribosomal RNA is it's a highly conservative structure, right? Because the ribosomal itself that they need this need when every time when you try to synthesizing something, right? And then
Yeah, yeah, it's
yes, yeah, you will need this,
important.
right? Hello?
Yeah, yeah, I get it. I get it.
Yes, yes. And so because it is very highly required in this, because it is highly required, yes, and you can see that it is highly conserved between different species of bacteria and archaea. And this is mainly because that yes, and because it is often, I mean, there are many genes, and for the genes that is very crucial and very key, and then their evolution rate will be much slower. You can understand that because if there are any mutation within this ribosomal RNA, and then this bacteria might just will die, right? Yes,
Oh.
yes. It is very easily to be understandable. It is very understandable, right?
Mm-hmm.
Yes. And then yes, and then so generally, yes, so generally that yes, and indeed, of course, that in the eukaryotic counterpart, we have another counterpart. Yes, and they have another counterpart called 18S ribosomal RNA. Yeah, yes, and but the 16S ribosomal RNA is the most largest one, the most popular one. I mean, it is about at least 10 times larger. I mean that means that if people do one, if people maybe do 10 16S ribosomal RNA sequencing, and then they might do only one, or I think we will even less than that, do 618S, yes, or ITS, we are for fungi, yes, and those are pretty rare, yes, and so the most common thing is 16S ribosomal RNA, and then because why they use this, it is because this it is just as you can see that it is not very mysterious. It is just a sequence. Yes. Yes. It is just a sequence like this. Yes. 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 It is just a sequence like this. A sequence like this, primary selection. It is just a sequence like this. And indeed, in this 16S ribosomal RNA, it also have many, it is about 1,500 BP, bit base. And then in this base, there are also many regions. They divided this 16S ribosomal RNA into 1, 2, 3, 4, 5, 6, 7, 8, 9, into 9 regions. And then the most common things they use is from V3 to V4. They just sequencing this 400.
3 and 4,
Yes,
V4 or
V2,
V3 and V4.
V3, V4, V3 to V4.
Oh, okay. But
Yes.
I have a question about this. This
Okay.
16 ribosome RNA gene is just one part of gene of the
Of the it's just a small part of all
yeah,
the
it's a
of
small
all the
part.
gene in bacteria in a bacteria.
Yeah. And in this small part, we talk more smaller. We talk just V3 and V3 and V4. So we just use this sequence to analyze this bacteria.
Yes. No. We do not try to use this sequence to analyzing a single bacteria. This is impossible. If we want to analyze a single bacteria, we need to do metagenomics. We need to exhaust all the genes within this bacteria, right? But
Yeah.
we use this for well, remember our title. Our title is community analysis of microbiota. What our purpose is identifying what this bacteria species, what the species of this bacteria belonging to.
Ah, okay,
Yeah,
okay.
yes. It is just like, for example, we are human. I do not need to test all your genes to identify you to identify you are human. If you give me a sample for a mouse and another sample for a human, we have many ways without exhausting all the genes. We can identify which is mouse and which is human, right?
Yeah, so our goal is just to generate the
Just to
phenol gene.
distinguish species. Yes.
So we
Yes.
just we do this job to generate the phenol gene. Is
Phylogenetic. Hello? Can you hear me?
hello. Yeah, I can hear you.
Yes. So our
Let's
general
continue.
thing is that we just using this very short sequence to identify its species.
Ah, okay. And
Yes.
I have another question.
Okay.
Uh, why we need to generate the phylogenetic?
Because it is a very easy way the basic principle of generating this phylogenetic tree is not very complex. It's just like if we comparing, we based on all the sequence, because we sequence every 16S ribose RNA there from V3 to V4, for example, that this 400 base BP 400 BP. If this 400 BP is very similar, and then we select it into very similar and then very close. If this 400 BP is highly different, and then it will be very different in this phylogenetic tree. So, yes,
Yeah, yeah.
yes.
So I mean, we get this
What is the
tree.
point? I
So
mean, you mean what is the point of generating this tree?
yeah, yeah, yeah. So we can use this tree to do what
There are
we
many.
do.
I mean, this is typically normally the first step we do that. Of course, you might wondering that, for example, maybe because such tree we can have. You can also download it online, right? Yes, because they even generating this phylogenetic tree based on multiple genes or many genes or whole genome, and then the result will be indeed even much more accurate than just using this 400 base. Yes, because if you as you can imagine that this because you're just using a 400 BP to identifying the species. Indeed, this the accuracy is not as you might think that accurate. I mean, I mean, if you analogous to it is maybe like it might distinguish wrong between a human and a mouse and a monkey. Yes, it is possible. But it won't identify wrong if between a human and a mouse. It won't. Yes. So
Okay.
because just yes. Sometimes you might identify wrong. The reason that we want generalizing this phylogenetic tree, I mean, I think normally this is the first step that first is for the computational reason because because in this computation we downloading the raw data and then indeed they have an intrinsic procedure that they need to remove the adapter. And then after removing the adapter, they need assigning all this because after they need to compare this 400 data 400 base 400 BP with the existing database because there is a database that tell us all the whole this whole 16S ribosomal RNA tell the sequence of many species they currently already identified this 16S RNA, their sequence. And then we compare one by one, compare our V3 to V4, this 400 BP, compare this to a database. And then if it match a certain level of similarity, and then we can say that, well, this is the species we want. This is the basic principles that for this taxonomy, because this is the basic principle that we identifying uh, which this sequence belong to which species. But however, there is an issue that I mean in this computation, I mean it is very natural. It is very natural if you're generating because you need to compare the similarity to I mean in the computation you need to compare computation to the species identifying which species you need to compare similarity, right? So it is very natural that you use such similarity data to generating a phylogenetic tree. I mean, because as I said, that if you have the similarity result from between, I mean, if you have a similarity matrix, you can generate it as phylogenetic tree. And I mean, when we try to compare this sequence to the taxonomy to the database, and then we need to what we also need to computing the similarity. So this is, I mean, it is just like when you're writing writing a software. Of course, if you're not generating the phylogenetic tree, the computation will be faster. But I mean, it is just like, well, when you write the software, if we adding this function, it will be it is just like we can also do that. So this is the first reason that why this is often typically the first step. And then I think another step is that by generating this phylogenetic tree that we can have some basic view of how complex our sample is. Because I mean, for example, if you sample this for a gut microbiota, and then and for example, if you sample this if you this biota, for example, adding many bacteria, this is for a gut that these patients just treated with many antibacteria, and then you can have a basic view that well, in this phylogenetic tree, this I mean, if you compare this phylogenetic tree to a standard phylogenetic tree, I mean, because we will have a relatively standard microbiome phylogenetic tree, yes. And then for example, if in that is just like according to your experience. So in this time, experience is very important. Then, for example, that like that, yes, like this is gut. For example, like they generating this phylogenetic tree from gut. And then if you, for example, if you're generating a phylogenetic tree, and then maybe you will see that, well, it is not as complex as it is shown here. For example, it shows like this taxonomy, this phylum, right? Yes, you can basically see that this is a very complex, yes, because it is I mean, basically yes, and all yes, and as you can see that this one step, and basically every yes, every species, I mean, possible in ranking in this, and they have a rank that, for example, like this four basic, this five basic phylum, it all contains that. I mean, if I mean, I'm just saying if you find that, well, there is one species that you can hardly find that, one kind of species, one phylum, one texture that you can hardly find that, or if the phylogenetic trees, I mean, generally, as long as it is not very like a typical phylogenetic tree, then you might guess either is the sample is sampling for a very unique situation, for example, like the antibacterial treated, or there are something wrong. There's just something wrong in this. But normally, if there is something really, there's something wrong, and then we might already test it because we have a quality control. We often, after we downloading the data, we need to first do a quality control. So in most cases, this quality control, we can identify that. Yes, but generally, if you do this phylogenetic tree, it is also helpful for you to have a basic glance, have a basic knowledge about how good this analysis is, the data quality is. I mean, because it is very obvious. I mean, if you look at many things that I mean, if you look at many data, you will have a basic. I mean, if whether this data is good or not. I mean, after you're generating this phylogenetic tree. I mean, because even the data pass the basic quality control, but there will still some differences in this phylogenetic tree. Yes, and so this phylogenetic tree that first we can identify. First we can have a basic view, yes, a basic view about how good the data is. And then is that we just because we also needed annotating the taxonomy, annotating the species, and then so we as a byproduct it do that. But it will require additional computational time, of course. Yes, is that all?
Yeah, yeah. Okay, okay.
Okay. And then yes, and then yes, and then after we generating, for example, after we processing, doing a phylogenetic, and then we do some prediction. Indeed because as you know that because we only know this 400 BP sequence. So indeed we need to do some. I mean, if in this case, I mean, if we learn something about metagenomics, that would be different. But I'm not sure whether we will talk because you can see the content here is already very large. I'm not sure whether I don't think it can be included because if just in 2 weeks. Yes, I mean if we don't do metagenomics and if but there is a way if you just based on 16S sequencing, you can use PyCrust. Yes, you use PyCrust. Yes, like this. Yes, and then this PyCrust is something that the basic principle of PyCrust is that if we have this, as I said that we have this 400 BP, right? If you have this 400 BP, and then you can search the related species, but in the database there is a because other people will they have someone that they already exhausted all the genes inside this bacteria, and then you can download it, the genes inside that bacteria, and then you can combining it to your sequence, and then after that it will form a database like that. I mean, so that means you can base on your 16S RNA, and then searching everything in a corresponding database that other people do already sequence the whole or overall genes in a bacteria, and then you combining their results, and then you can form a architecture that human-made metagenomics. The metagenomics is something like that. We sequence all the possible genes inside the soil. Yeah. So you can also form a pseudo sample, but it is a pseudo sample because it is we downloading other people's data to generating that. This is PyCrust. Well, this is PyCrust. What PyCrust did, and they developed this in yes in many nearly 10 years ago. Yes. Uh, yes. And then this is yes, and now currently or nearly the second generation, and if we using this PyCross, and then we can predict it, all the genes inside the soil, and then you know that each genes can corresponding to a enzyme, right? You know that.
Yeah.
Yes, and each gene can corresponding to an enzyme or anyway, it is a protein. And then for each protein, they have their corresponding metabolic pathways. They catalyzing, normally catalyzing something, right? Yes, and then so in this way, we can predict it, the metabolic pathways within these microbiome communities. Yeah. So and then for each metabolic pathways, they often they have different functions responsible for different functions, and then you can also do some prediction. And indeed, this PyCross, it is not as difficult as you think because PyCross already they have a very good integral, and then you just need one just one line code to do that, and this is in Python, and then so. But I just told you the basic principles, and they will do that. And yes, and you can very quickly generating. It will require about. It depends on your computer. Depends on your computer. And then this is functional analysis. And then we have the microbiome ecology. And then microbiome ecology is something like interaction within microbiome communities this is something more computationally based. Yes, but I mean microbiome roles and ecosystems and their interaction with environmental metabolites. This is something more computationally that means, for example, this is for example, if we have many species, and normally we can expect it that because there are some species that will uh, that will suppress that is harmful for other species. Some bacteria are harmful for other bacteria because they can excrete some just like antibiotic. It was extracted by bacteria, right? Secreted by bacteria. You know that, right?
Yeah. Yeah.
Yes, yeah. So some bacteria is harmful for other bacteria, right? Yes, and in this way that indeed we can. There are some computational method that we can identify such interactions. Yes, we can identify, for example, what normally if there is a bacteria that is very harmful for other species, that we have some algorithms that we can detect that. Yes, there are, and then we will introducing those algorithms, and also that introducing those algorithms, and then their basic principles and how you use that. Yes, and we will introduce that. And also another thing is that the microbiome role in the ecosystem. This is so first is the interactions within the is bacteria interaction, and then there are some because microbiome that itself it not only because for example like it excreted antibiotics, and then antibiotics it is a metabolics, right? It is a small chemical compound, right? Yes, and then that means indeed every bacteria it constantly excreted some metabolites, and then it will also absorb some environmental metabolites for its usage. And then so we will basically discuss that how we can explore the roles of the microbiome roles in ecosystems and their interactions with environmental metabolites. Yes, we will talk that. And then I will discuss that some basic principles, and because there are a little bit differences between this and in their computation, and then we will discuss that. And then is some, for example, some animal, and then I will discuss because in our course that we will use animal microbiome. It's mainly from gut. But indeed, the animal bacterium microbiome, their current study, they even study some face everywhere. As long as you think they can find, they already study some microbiome. Yes, but we will focus on gut. And also plant microbiome. Plant microbiome is it depends also there are some leaves or fluid or rhizosphere. It's the root microbiome. Yes, and we will mainly focusing on root. Yes, on the rhizosphere. And also environmental microbiome is soil, water, and extreme environment microbiome, and then this and also the impact of environmental factors on microbiome communities. Yes, and also generally we will use these three examples. Yes, we will download some examples, and then you can download some examples to do this analysis. Yes, and so the basic thing is like that, and then you will have a deeper understanding how these microbiome structures have their distinct structures. And then I will discuss something about, for example, some this is relatively literature, and then for example, currently our microbiota research, and then some trend. Yes, and then I will briefly introduce that. Yes, and then this is, and then I mean, of course, there is something. I'm consider whether we can teach metagenomics, but because we only have 2 weeks, I don't think you can see that this content is already relatively intensive, right?
Yeah.
Yes, yes, yes. I'm not sure whether we can teach all this uh, all these things in 2 weeks. So if we're adding another metagenomics is something that we sequence all the genes inside a sample. Yes, and then so the all the and then assemble it, and then so we can know that, or for example, like this is answer your question. You can study all the genes inside a bacteria. Yes, yes, it is called metagenomics, genomics. But this is much more expensive as you can imagine. Yes, much more expensive than 16S. Yeah, 16S because it is very cheap. It is super cheap. Just it depends. It depends. I mean, it just generally just less than 1,000, less than 100, less than 100 yuan for one sample. Yeah, so it is very cheap. And then our basic assessment is that first, I would let you to write some brief summaries, and it is you might find a. I will further discuss that you might find something, and then, for example, like if you're analyzing an animal microbiome, and then you need to write a basic summary for that, yes, for that, and then yeah, and then it's case study. Uh, and then also this basic summary, this case study is that you will need, for example, I mean, as I said that I will give you some example, right? You need to download the online data, and then and then in that case I mean, generally I will read your analysis result. You do understand? I mean, you downloaded some online data, and then you're processing all this analysis. And then you will wrote a brief summary, and then I will read, and then you will need to wrote a brief analysis report. Yes, a brief report analysis report about that. So this is case study. Yes, and then analysis report and also the related things, and then this is checking your understanding. Yes, and then of course, we have a bonus that if you teach, for example, you are good in Python, right? If you teach, for example, other students, I'm not sure whether all the students in this course they took this introduction to computing with Python. I'm not sure. Maybe someone haven't. Maybe someone haven't. Right? Yes. If someone haven't took this course, and then they if they have any difficulties in storing the Python, in installing the Python, and also because there are many people here, maybe someone using Mac, someone using Windows. What kind of platform you choose?
Windows.
Windows, okay. Okay. Windows, that would be less. Okay. Anyway, that if you can help your students, yes, help your classmates, and then teach them, and then yeah, there is a bonus. Yes, there is a bonus. I mean, yes, and if you install this, I mean, for example, like the PyCross was in. I mean, I'm not sure whether they have any issues in installing that. Yes, and if you help them, it can be a bonus. And then also that, yes, there is a bonus. And also that if you do, for example, not just one case study, if you do more case studies, and then it can be a small research projects. Yes, I mean, you download it, other people's work, but you're reanalyzing it. It can be a small research project if you very carefully, and I mean, so it is just a higher level of case studies that if you study very well in this analysis, and you can do a relatively mini research projects, and it is also a bonus, and we can even consider to publish that. Yes, and yes, we can even consider further publish that, and then you can be a co-author in that paper. Yes, okay.
Okay.
Yes. And then this is yes, it depends. It depends. I mean, generally it is a higher level of case study. Of course, you need to so you of course you need to first do this case analysis. Case studies is generally that you pick maybe 6 samples. Yes, no many samples. Yes, and then for a mini research projects, of course you need more samples. And then it of course if you also need to have the time. And then, of course, and then it's our course policy, and there is no attendance requirement. for this course that of course that I mean I just generally but generally I need to say that if you don't attend then maybe maybe you might lose the schedule, yes. And then because I mean you can see that our teaching is relatively intensive, yes, intensive. I mean if you don't come, I'm not sure whether you can catch up. Yes, I'm not sure. Yes, and then but anyway, I won't anyway that I won't check that whether you attended or not. I won't do such things. And then is that students are expected, yes, and then also this case study, I mean, it depends on because for someone they might be difficult to do a case study by themselves, yes, and they can work with their classmates, yes, and for example, do this analysis, they might working together. It's okay. I mean, if you do, I mean, I expect many people might need to work with three or four people together to do a case study. It's okay, and then. But you need to say that. I mean, whether I mean what kind of we can form a small chat group, and then I can know that basically what kind of job is done by who, right? I need to know that. Uh,
So if we work in group, we need to have a small group, and you need to
I
know
can I
who.
can form. I mean, if you want to form a group, and then you can tell me, and then I can form a WeChat chat group. Yes, yes, and then
Yeah.
and then you just discuss there, and then I will know that basically which part was done by which, right?
Okay. Okay.
Yeah, yes, 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 yes. It's like that. I mean, yeah. I mean, generally, I need to know that which part is done by which, and then one starts at, and then this, because I expected that it can be start tomorrow. I mean, or because I'm in Berkeley, now it's 14. But oh yes, it is already 15 in China. But I mean, it seems that today there is another people who you are 那个姚志标 are you listening here? Okay, it doesn't matter because it seems that today we don't have many people. We at least now we only have two people here. I mean, so I expected that now most people are still taking their course, taking their final exams, right? Like you said you also have several B. You need to talk B,
Yeah,
right?
I have two B exams.
Oh, yeah. So this is an issue that because the A is finished today, right? Yes,
Yeah, yeah. A
yes,
is
A
finished
is already finished, and
for
so
every student.
yes, and so if all people need to talk B, I mean at least now it seems that it is the case, and then well we might have to postpone our schedule. Yes, 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 postpone. I mean, because I mean if this course I mean to you is just one people, but if every course I mean is just you also I mean. Also, you also need to talk B, right? I mean, you don't
Yeah,
have the
yeah,
time. Yes, you don't have the time. Yes, and so yes, I mean because my original expectation is that there might be 10 students in the first row, and then maybe 30 students in the second row. Yes, my expect is that. So generally, let us see. I mean, how many people can finish? Yes, yes, yes. Okay, how many people? Let us see how many people can finish within this week. I mean, so if we think this week there can be some people finish, then we can start maybe start in this week. Yes, with as long as there are 10 people, I think it's enough. And if there are less than that, we might consider to combining. I mean, just yes, yes, yes. We may be because we have another diabetic neuropathy study. We can first finish another paper and then start this course. I mean, maybe just study at the end of the July. Yes, yes, and then
yeah, yeah. Okay.
yes, yes, yes. And then if we study at the end of July, and then we might, I mean, the pace, we might try to make it slower because we need to make sure that most students in this course they can do at least they working with other people. They can finish a case study. Yes, 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 yes. That is most students pass this course. Yes, yes, this is. I think at least 80% can pass this course, yes, and so we will generally control this pace, right? Okay.
Okay, okay.
Yes, yes. And then
And the summer semester will begin on July 29th.
29.
Yeah, July 29th.
Wow. Oh, so it is. If you have any other summer courses, yes, it might. Yeah, but I think it's okay because I mean, even during the spring, you also have a lot of courses, right? Yes, and then
Yes, yes.
yes, and
But I
then I
mean,
mean,
maybe the
there
course
are
is,
some biology classmates that some students just like this teaching assistant, they are from biology. Some are from
oh,
biology, yes, and they have some courses. I mean, because every day, I mean, I expected that you spend 2 hours is enough, I think. Oh, yes.
okay, okay.
Yes. I mean, 2 hours is that maybe 1 hour in the class, 1 hour after class. I think is enough. Yes. Yes. I mean, if you yes, of course. I mean, if you spend more time in teaching other people and doing more projects, and then you can have bonus, and then you might even publish the result. But if you don't have much time, I think it's also okay. Yes. I think 2 hours is enough. Yes. Okay.
But the time I think should be at the evening, in the evening.
In the evening. Oh,
Yeah,
in
because
the evening. Okay,
we
okay.
have
It's okay.
school.
Yes, it's okay. Yes, it's okay. Because the evening is I need to wake up at 6 or 7.
Oh,
Yes. Yes, it doesn't matter. It doesn't matter. I can wake up at that time.
okay, okay.
Yes.
So where are you now?
Okay. Thank you. And then thank you for
Oh, thank
listening.
you, thank
Yes.
you.
Yes. Bye. I need
Bye.
I need to have a dinner now. Yes. 那个还有姚志彪do you have any questions? Hello? Do you have any questions?
Really?
Uh, you don't have any questions. Okay, so we end. Okay.
Okay, okay.
Uh,
Goodbye.
can I just listen to the lesson? Yes, of course. Yes, of course. Yes. It doesn't matter if you don't talk any case studies. You just listen. It's okay. Yes, it's okay. You do it. I mean. Anyway, that in our course we don't have any mandatory. You can just listen, and then I mean because you might be busy, right? Yes. You can just listen, and then after that, yes, or not do anything. It's okay. Yes. Or I mean, anyway, that we don't have any specific requirements. We will see that which people they really do the case analysis and or can finish the mini research projects. We don't have any requirements for other people who are because for example if you are very busy, you don't want to do that, or after that it's okay. Yes, it's okay. And you don't need to leave this group. You don't need, and then you can just watch that and listen. Yes, it's okay. It's completely okay. Yes. Okay, and then I will have dinner. Thank you. Yes, thank you.